Alright, so taking a look at it from superficial to deep, we're looking at the layering of the muscle. We go from that fascia that we mentioned just a second ago, that fascia which is going to give rise to the tendons. As you can see, it starts to stretch out into the tendon, which then becomes part of the bone. Now, if I'm going to work my way down from the muscle to the fascia, that whole muscle is going to be broken down into muscle bundles or muscle fascicles. And before I can actually get into the fascicle, which is shown right here, you can see that the epimysium is going to be the outermost cover and surround of the whole thing. So that's shown there as the epimysium. Now, each individual bundle, that fascicle, is going to have its own connective tissue around it, and that connective tissue is going to be the perimysium. Now, that individual fascicle is going to be broken down into these little dots there, which are individual muscle fibers. Now, surrounding each muscle fiber is the outermost connective tissue. The outermost connective tissue, which is the innermost connective tissue, which is the endomysium. And then if I break that down even further, inside the muscle fiber there are individual myofibrils, and inside the myofibrils are these two filaments. Those are the most important ones that I need you guys to know. If I'm going to zoom in real quick on those, you can see that one is thicker than the other. This thin one is actin, and the thicker one is myosin. Those you need to star and need to know as we continue on, because as we look at it, you guys can see kind of the lines in between there, those lines that are within there. That is the space that you start to see the striation. So we actually see the Z line to Z line, which, or I should say, the sarcomere, but we're going to see a contraction that's actually going to take place between those. Now, if we did a study now, as you can kind of think of this possibly an extra credit opportunity, and it doesn't have to be pencils. It's just taking something like a straw and having paper wrapped around it. That paper wrapped around the individual straw would be represented as the endomysium. Now, if I take multiple straws that have their own paper around it and wrap it up in a different type of paper, that would then be considered the perimysium. That whole individual thing would be considered a bundle. So now if I have multiple bundles and I strap another paper around it, that would be considered the epimysium. And I do one more paper around all of that, you would then have the fascia. So this is just kind of a study analogy to make sure you understand it from superficial to deep. So it goes fascia, epi, peri, endo, and then we're in the myofibrils with the actin and myosin filaments. Now, when I was just mentioned just a second ago, you can take a look at the Z line to Z line, and that's this illustration, this strand there to that strand there. That unit is called a sarcomere, and in there, this is the actually contracting unit. So from there to there, there is a space in between here. If you guys can see that space right there, that space is going to be important. That is actually known as the H zone. That H zone actually will go to 0 during a muscle contraction. Now, we're not going to get into a lot of it, but it really comes down to is that there is, being that this is a voluntary muscle, we have to somehow have our brain control the operations of it. Ultimately, what's going to happen is we're going to send signaling down through the nerves here, and it's actually going to get into what we call the sarcoplasmic reticulum, and inside there, stored in there, are particular minerals inside the bloodstream of it that can actually stimulate the contraction of these two filaments, and those two filaments are the actin and myosin. Next slide, you start to see it. Now, in order to have the particular nervous system work with that of the muscular system, we have to have a junction point. And a junction point is also known as a meeting point. So we have a neuro, which refers to nervous system, and muscular, obviously the muscular system, junction, in which the brain, or in particular the impulses sent from the brain, are going to meet that of our skeletal muscles for a voluntary movement at a particular site, and that's a junction site. And so when we're dealing with neurons that we can help control, we send out signals, we're going to be working with what we call a motor neuron, because it's going to send out information to do some sort of action. And then the muscle fiber obviously is what is going to control. Okay. Now at the end of the particular neuromuscular junction, you have what's called a motor end plate, and at that motor end plate is where we are actually going to see the particular process taking place, where we're going to see chemicals actually being released, and those chemicals are called neurotransmitters. So here's an illustration where we see this guy right here is the end of a particular motor neuron that's shown right there. And so that is the motor end plate, the bottom of it, or we can consider our neuro, and this down here is the muscle, so muscular, and that's the meeting point, so it's a neuromuscular junction. And notice on this particular motor neuron, there are actually 4 junctions. There's 1, 2, 3, 4 different junction sites. Now, each of those has its own individual meeting point, which is helping to control each individual sarcomere, which is located below. Okay, however, they're going to help control that. Now, this entire thing, if I'm going to highlight and circle this entire thing, that whole process, not just the junction site, but the whole motor neuron, the entire muscle fiber it controls, is called a motor unit. So the entire motor neuron and the muscle fiber that it does control. Now, what it's stated is that it's an all or none response, meaning that if you want to do a contraction, you can't just kind of do it. Either you're going to have actin and myosin flip or it's not. Now, the easiest way for me to kind of illustrate that and have you mentally picture that is a mouse trap. Once a mouse sets the trap, does the spring just go halfway and say, yeah, you look too nice? I don't think so. The spring is just going to go. As soon as it hits the thing, the stimulus starts, and then the spring pops into action and snaps shut. All or none. Once it starts, it's going. And that's what our muscles do, as well as our nervous system goes through. All or none responses. So as you can kind of see, the whole illustration here that's going to be taking place, kind of get rid of all of that. But in particular, what's you want to look at is at this site here. So this thing is the motor neuron. So inside here we're having an impulse being sent down from our brain, and inside there these are vesicles. And you learned in the first chapter vesicles are nothing more than a sac of phospholipid bilayers, and in there are tiny little chemicals. Okay? And in those tiny little chemicals are signaling structures. And those tiny little chemicals for this particular unit is called acetylcholine. Once the neurotransmitter releases the chemicals, the cell itself exits through exocytosis, and then we start to get these little chemicals that are going in this little junction site. And that junction site is known as a synapse. Okay, a synapse. And as soon as those release into that site, that stimulates, this chemical stimulates the next impulse within inside the muscle fiber to take place. So as it continues to go through, if I'm looking down here, notice that there is a muscle fiber below. So this is all the muscle fiber below here. The chemicals are going to be released in that, which stimulates then the next part to take place. Okay. Now, how does it work then? What is taking place to cause a muscle contraction? And ultimately what a contraction is the shortening of a muscle, and shortening by sliding actin and myosin together. Now the ultimate thing is this is voluntary. So being this voluntary, your brain has to control it, hence the neuromuscular junction. Our nervous system has to be in line with that of the muscular system for it to work properly. Now another thing that is needed is energy. Energy in the form of ATP. And we recall that in order to make energy, you must have glucose as well as oxygen are the primary energy sources for our body. Now, where is it made? It's made in the mitochondria. So if our muscle cells need a whole bunch of energy, they better have plenty of mitochondria there so that they do not get tired out. As I mentioned ago just a second, that there is one chemical and their neurotransmitter which is going to be released. So if you go back here, these individual little black dots that I have there are those chemicals, and that chemical you need to know, highlighted star, is what's called acetylcholine. Acetylcholine is the chemical that will be released at the neuromuscular junction, and it will stimulate the next step to take place. Think of that as the start of the mouse trap. This is the cheese that has been placed on, and now the mouse has got there and it's about to touch it. Now, what happens once that cheese is there? In comes a mouse. Once that acetylcholine is released, in comes calcium. Calcium is being released actually from the muscle fiber. It leads into the muscle fiber from the sarcoplasmic reticulum. Now that muscle fiber Once it receives that calcium, in comes our action. This is the mouse hitting the trigger switch, and as soon as that calcium is there, actin and myosin slide together. Now here, you guys, if you can take a look at this structure there, that structure there is called troponin. If you can see it here, it's the troponin right there. Think of that as like a key. And if you also picture that, it looks kind of like DNA, twisted ladder. Think of it as a twisted ladder. It's locked, and there's the key. Somehow I need to open up that key. And the way I do that is I add a particular component to it, and what I'm adding is that as soon as calcium lands on these keys, our ladder untwists. So it goes from that to a nice straight ladder to get hooked up onto. Okay? Now, calcium again. Calcium is going to bind to that particular site. What's going to do is it's going to untwist, and then these hooks that are down in myosin will actually hook onto actin, and as it hooks onto actin, it slides together. So if you kind of picture this, what's going to take place is that these guys are going to hook on, hook on, hook on. Okay, as it hooks on, this space is called the H zone, all the way up and down. These bars of the myosin will slide them together, and that zone goes to nothing. So as I shrink that zone, that shrinks the muscle cell, which causes the muscle to enlarge in a contraction, which causes the movement. Okay, so ultimately, what took place? What took place, so you get any kind of a step-by-step motion of it. Let's add a slide here. Step one, what we needed to redo is have at the neuromuscular junction release acetylcholine. Okay, acetylcholine is released at that neuromuscular junction, which then stimulates step two, calcium to be released. Once calcium is released, it attaches to troponin. You do not need to worry about troponin, but it attaches to the actin filament. Calcium binds to actin, which causes it to untwist. Step three, myosin slides together, and as it slides together, it causes the contraction. Now, right now, myosin and actin are hooked together, and they are not letting go. They are locked in. The only way that I can actually break the actin and myosin bond is to actually have this guy happen down here. I need ATP. So one of the things that we talk about is ATP being needed for energy. Absolutely. But ATP is not necessarily needed for the contraction of muscles to take place. The contraction can happen, but the relaxation cannot. Okay? So when we go into our steps, in order to have a relaxed state, we need to have ATP actually break the actin and myosin apart from each other. So ATP must be there. Not only that, if this was step one, acetylcholine is released, it's going to stimulate the contraction to take place. So if we don't want that contraction to take place, we better get rid of that acetylcholine. Step two, if the key is to unlock the troponin to make sure actin opens up, and that key is calcium, we need to make sure that that key goes away. So we want actin to lock back up. And obviously we want actin and myosin to slide apart. And what I need to allow that to take place is energy in the form of ATP. Okay? So any questions if you guys do have on those, please make sure you write them down. We can go through this again multiple times until we get this. The next PowerPoints after this are going to be looking at the actual individual muscles as well as the skeletal muscle fatigue and how over time, based upon these relaxed states, our muscles do get tired.